Okay, hi, my name is Linnea.
I'm Bezia.
And I'm Linnea, and this is episode number 4 of the Lemma Journal Club, the show where we read every paper to let you know if you need it. You probably do.
Where if you need to read it.
Need to read it.
Because they don't need the paper. They need to read the paper.
No, they need the paper.
They need the paper. Okay. We have a terrific paper for you today. We are talking about the research paper, Dramatic Changes in Mitochondrial Subcellular Location and Morphology Accompany Activation of the CO2 Concentrating Mechanism. It's by Justin Findenier as the first author and Arthur Grossman as the last author. Published in PNAS in September 2024. It's another mitochondria paper. We might secretly be a mitochondria podcast. Why did you pick another mitochondria paper?
I chose this paper because of the mitochondria and chlamydomonas. So Bez and I have been on a long quest for the past 4 years of our postdoc to find a joint project, and this was the first project I heard about, or the first other project I heard about that I thought, oh, maybe this is something Bez and I could work on together. Right. The first author gave a talk maybe 4 years ago at Princeton that I thought was very intriguing and was fascinated by the rearrangement of the mitochondria.
Yeah, I was thinking before this, I was like, oh, we have some data that we took on this that I could go bring up, and I could do a little thing and just check a theory I have about the paper. And of course I didn't do that. I wonder if the supplementary video to this is going to be those time lapses that we took, not that they took, but that we took of some of this.
Yeah, we'll have to go back and look at what those look like.
So now for a segment that I'm going to call Speed Read. I'm going to summarize the paper with a reading of this significant statement as quickly as I can, so that if you didn't read the paper, you know what's going on. What do you think of the speed read?
Yeah, go as quickly as possible.
I should say before we get further into it, if the audio is particularly bad or good this time, just know that every time we record this podcast, we do so with a completely different recording setup. And today I am holding my microphone manually. So.
And we have construction going on in our house.
Okay, so here's my speed read. Mitochondria often form a dynamic membrane network that can change their intracellular location to sustain adenosine triphosphate, ATP, consumption for specific functions such as synaptic vesicle mobilization in neurons or photosynthesis in plant and algal cells. In this study, we investigated changes in mitochondrial ultrastructure and its subcellular location in the green microalgae clammy and demonstrated the role of tubules to achieve mitochondrial reorganization when the cells experience very low CO2 availability. We also gained further insights into the functional importance of mitochondrial respiration and carbonic anhydride in the context of CO2 concentrating mechanism, and raise questions about how mitochondrial dynamics can respond to the diverse energetic needs experienced by photosynthetic cells. If you didn't get that,
very
it's a
speedy
podcast. You can just play it again and again and again.
Or slow it down.
Yeah. 0.5x. Make it sound choppy. I will also say, Justin Findenier and Arthur Grossman, if we are unusually, or if I am unusually negative on this paper, it has nothing to do with your paper, even remotely. I got some bad news about some of my own science earlier today, and now I'm just a negative Nancy in general. So don't take it personally. I'm just jealous.
Having a paper out in the world.
Yeah. I found this paper super dense. There was a point at which I felt that figure supplementary S7A really should have been in the main paper. And I was like, look, this is PNAS. The page limit is 12 pages. You could put these figures in the main paper. The paper is only, and then I scroll down, the paper is a 12-page long paper. They hit the PNAS limit.
You did text me and say, this is a really long paper. And I was like, I don't think it's that long. I was reading through it, and I was,
They've hit the PNAS limit, right? So, they actually did have to bury a lot of things in the supplementary. They made a choice, essentially, to make the figures that they do have really big.
Really big, yeah.
And that's, that's their choice. And as a result, some of the really important information does go into the supplementary. Yeah, to me, this was a huge paper.
This was
And we're recording a day late, so I would argue, even though you say otherwise, it was a huge paper to you as well.
Sure. This was the first paper that I read. So I read part of it on the PNAS website, and scrolling through all of the supplementary videos through the paper. So PNAS puts the supplementary videos in line. And I actually found that very distracting in this paper.
Oh, yeah, I love it. I love it.
I didn't like it here, but that was the first time I felt that way.
Okay, so kind of diving into it, right? Like the main thing here, here, let me ask you a question.
Yeah,
What did they discover in this paper?
So that I was unaware, because when I first learned about this work, I was brand new to the clammy field. So I was unaware that it was already known that mitochondria relocalize during CCM activation.
Right. So
I thought that would have been the major discovery here. But that was...
I'm just going to say, for people who have not read the paper yet, the CO2 levels go down, the carbon concentrating mechanism of the clammy activates, and the mitochondria move to the periphery of the cell. And there's a bunch of pictures of that in this paper. But that was already known before this paper.
That was already known. It was known through TEMs, I think. So probably only just barely known.
Okay. So maybe this solidifies.
So I think this definitely solidifies. But then beyond that, I was hoping for a little bit more insight into why this happens. And they do try to do some molecular dissection, but the link to function, I think, is still really unclear. And so overall, I was a little unsatisfied at the end of this paper.
Do you know roughly, are these people, it reads like it's by biologists.
Yeah, so Arthur Grossman is a very established Chlamydomonas biologist. I think he's retiring now or just retired. And Justin must be a biology PhD student.
So the obvious thing from the get-go, right, and I already was putting comments in this on the paper in my own notes from the very beginning, is that, the mitochondria are moving as a result of the CO2 concentrating mechanism coming up. The clammy are pretty small. How big is a clammy?
10 microns.
10 microns. One wants to know, is diffusion extremely slow of CO2, of oxygen, of ATP in a clammy? If it's not very slow, then you really have to scratch your head at why is anything bothering to reorganize. If it is really slow, then you make a spatial reaction diffusion model to see, what are the effects in the very well understood chemistry.
Especially in a PNAS paper, a modeling section would have been really beautiful.
And so I assume it's just not who they are.
Yeah, I don't think so. And Arthur, I think, was retiring during this whole writing this paper, so I'm not sure he was branching out in new directions.
Okay.
But that's
But it seems to me like one could take essentially the patterns shown in this paper, take reasonable guesses at most things. The one thing that I'm kind of iffy on is what is the diffusion, because I've seen TEM of clammy, and sometimes it looks like they're to try to reduce CO2 leak and stuff like this, they really, really pack themselves in. So maybe actually ATP moving around is an incredibly slow or very directed process, and that's why you have all these little tubules going on inside the cell. But that's what I want, is, I rarely say this, but just someone with a little bit of computer ability to write a very small theory paper showing the reaction diffusion of why certain things might end up more efficient if your mitochondrial respiration was on the outside of your cell in low CO2 conditions. I think this is an easy layup. It's literally 3 days of work.
Yeah.
That's what I thought the whole paper. I just kept thinking that to myself over and over again. The other thing is, your clammy people really like their acronyms. Can I throw some acronyms at you and we see what you know?
Sure.
LCIB.
Low carbon induced bestrophin. What does B stand for?
What is bestrophin? I couldn't find it in the paper. I tried to Control F and I was they've dropped this on me without defining it.
Okay.
So I'll use it in a sentence. Also, at VLC, the CCM protein, LCIB, encases the pyrenoid, potentially capturing CO2 that leaks from the pyrenoid, converting it back into HCO3, which is bicarbonate. So the CCM protein, LCIB.
Yeah, I know that it's a CCM protein, a low carbon induced. Maybe it's just low carbon induced protein B, but it could be a bestrophin, which is some sort of bestrophin.
Can I give you some more?
Sure.
What is LL and HL in the context of CAP?
Low light and high light.
Low light and highlight. Oh.
Yeah.
Okay.
Yeah, this did feel very like clammy specific, so I do apologize to Bez for making him read it. I really thought it was going to be more mitochondria. I do have a lot of like things.
There's a ton of mitochondria. There's a ton of microtubules. There's the miro protein, which made me really happy to read a paper where they're doing stuff to the miro protein.
I think a lot of interesting things that we've been talking about of the energetics within a cell and the mitochondria chloroplast interactions, I think, are super interesting. But the paper was definitely written for the clammy field. Like, you can see this in their use of acronyms and also in the conditions they probe.
Yeah, I was able to like get through most of it. There's like lots of little nitpicks that I could ask you questions about, like this LCIB and cherry channel is supposed to tell me if the cell is acclimatized, and then in a lot of the data it looks like it's not acclimatized. So why are you taking data before it's acclimatized?
Well, so I think this is one of the secretly interesting results of this paper, is that the field uses low CO2 as a CCM necessary condition, but a lot of their data suggests that you have to go to this very low CO2 levels to really get full induction of the carbon concentrating mechanism. And yeah, there's some discussion in the labs about, bubbling versus stirring versus, and these are like, you say in your papers what you're doing, but how can we compare experiments across labs is an ongoing discussion. I got the idea. This is again going into the words. I'm just kind of going through it in order. The word sparging. Yeah, sparging. I never heard this word.
Oh, you have never heard? This is not from your field?
I meant to Google it.
This is it must mean like blowing gas to shoot out other gas.
That's always bubbling in my sparging.
Oh, is that what you mean when you say bubbling? Is to blow out one gas with a different gas?
Oh, no. I mean like you take CO2, 6% CO2, and you pressurize it so that it goes into liquid media and bubbles. You're saying sparging is like when you try to back like your DMSO with nitrogen or something?
Yeah, that's what I thought we were using the word sparging for. But they do say sparging cells with VLC and sparring cultures with HC or LC. So,
okay. I don't know what sparging means. I was
just such a
I meant to Google it and I totally forgot.
It's such a good made-up word. It's like a bleb.
Nobody will give you funding for sparging.
What is the one to like steal apples in the UK?
Oh, yeah, from Gavin.
Yeah.
I forget.
Oh, well.
Yeah.
What do they mean? They try to talk about the geometry a lot in this paper. And the geometry is consistently confusing the way that they talk about it.
Okay.
They try to use the words apical and basal here.
Yeah.
What is the apical side and what is the basal side of
So I am not like a great person to ask because I don't understand what apical and basal means in regular biology, but a clammy cell has like an orientation.
It's a front and a back.
It has flagella.
It has flagella.
And so I think that determines is that going to be the front or the back?
I don't know, but
that determines one side.
Okay.
And so there's flagella, there's the pyrenoid, and there's the nucleus, and they all align along one axis.
Okay. They tried a couple times to describe what direction things were going, and I read that carefully, and I had an idea in my head, and then I got to figure 2E, and I was like, oh, this looks nothing like I understood their description.
Oh, really? Okay. This is the result that I saw several years ago and was like, wow, how beautiful,
this is like, the 2E is like super cool.
Yeah.
But their geometry words did not
did not vibe with your.
Yeah. And so it's particularly like 2E, the maximum projection looks super different than everything else they're going to show, where they're like, oh, the mitochondria go to the periphery in the very low CO2 conditions.
Yeah.
But like, they don't just go to the periphery, they go to the periphery in a manner that looks like a circuit board or something, right? And so when they do just the like 2D slices inside, you see a very empty cell. But when you're on the periphery, you see this beautiful circuit board.
Yeah, I guess I wouldn't use circuit board as like so much as envelope or like.
But like, look at the way it moves. It looks like literally one of these like.
What do you mean moves?
No, I mean like, you know, look at the way the lines go. It looks like the etchings of a circuit board that you would have like on a motherboard or something, doesn't it?
I don't know. It doesn't. It's not reminiscent of circuit boards to me.
Oh,
but
really?
It's so beautiful, and I'm still like, why on earth does this happen? And this is one of the questions I had really hoped would be answered when I read this paper, and it wasn't. And I think they tried really hard to answer it, but
I don't know. I feel like they did the thing, and this is going to become a trope. I gotta like come up with a snappy way to say this. They did the thing that every biologist does, which is that they tried 18,000 inhibitory drugs.
Yeah.
And saw what they did. And look, at one point, they destroy all the microtubules.
Yeah.
And all the actin.
Yeah.
In the cell. And they have the gall to say, when both drugs are present, the effect is surprisingly severe relative to the loss of individual cytoskeletal components. No shit. You just destroyed everything in the cell. There's nothing there that could rescue anything anymore. What do you mean surprisingly severe? All they've got is hammers.
Well,
you know.
but I like, so they show they have this one mutant or drug, I forget. It's figure 3B, where they are able to only mess up the orientation of the mitochondria and not the. And this also happens in Miro, right?
Yeah, so this is the NAP11 mutant, which
the NIP11, NAP11.
I think that's 3B. It says
3B?
in the thing.
No, that's 3A.
3A, effect of lanolculin.
So that's getting rid of actin in the NAP.
Oh, you're right.
And then I think B is getting rid of microtubules somehow through this APM.
Yeah. Which, had you heard of APM before this paper?
I had not. So
yeah.
I do find that it's a good tool to have around. But I think...
a tool. Is it a good tool? Like anything that like APM tech, like whether you stabilize the microtubules or destroy them or whatever, this is such a huge perturbation.
Yeah, it's a huge perturbation, but if you're worried about, so like I'm in the condensates field, if you're worried about your condensate size being limited by the microtubule network, if you destroy the microtubule network and your condensate size doesn't change, that indicates that the microtubule network isn't physically constraining your condensates.
But I'm not in the condensate field.
I'm just saying that's an example.
I'm interested about the mitochondria. They mess with the microtubules with the mitochondria.
Okay, what did you think of the Miro mutant
What? Okay, so like Mirror Mutant is a great idea. Obviously, we should do it. Obviously, I'm trying to do it all the time. At some point, they say that, the mitochondria still kind of get where they're supposed to.
Yeah.
Even when they've inhibited Miro. The quote is, inactivation of the Miro1 gene dramatically impacted the parallel arrangement of mitochondrial membranes, but the mitochondria still relocated to the cell periphery upon exposure to BLC.
Yeah.
Like, I get the impression sometimes that biologists think, or that somebody here who just thinks that this is a flow diagram, right? And like they've inhibited this thing, and it turns out not to be connected. Like, if the Miro isn't there, how are the mitochondria moving? How are they moving? How are the mitochondria moving?
They are moving, though. They observe them moving. Like, look at 3C.
No, my interpretation of that is that something rescued. Right.
Something rescued the
There's another
Miro.
linker that, like, this is a kinesin 14 versus dynein situation again. That
but
moving mitochondria is so critical that there are many ways to rescue the motion of mitochondria. That's my interpretation of that. That their inhibition of Miro1 failed.
How do you reconcile that with the APM? with B, where the microtubules are gone and the mitochondria move to the periphery,
Again, rescued.
rescued by actin.
Yeah, I
But
guess
isn't
if you're
in
able
the
to kill
mutant?
both, like Miro is supposed to be a linker between kinesin and the mitochondria. It should have moved it along the microtubule network to where it needed to be. If you can get rid of Miro or you can get rid of microtubules, it implies that there is another way for mitochondria to get where they need to be. Could be actin. They say that clammy don't have intermediate filaments. Not that I thought it was going to be intermediate filaments. Could be, I don't know, some sort of osmotic pressure, treadmilling. I don't know. Maybe you can purl really quickly, propel yourself that way. No,
yeah.
just to be clear, I'm joking. We're smiling at each other right now.
Okay, so some other mechanism. That's fine.
Is it fine? I mean, that's wild. What is the other mechanism by which mitochondria get around?
Wiggling.
Wiggling. I do think I heard from somebody at a conference once that there is an actin based method for mitochondria to move.
I think B is they've knocked out actin, and 3B is they've knocked out actin and mitochondria. That this is in the actin.
Actin and microtubules.
Oh yeah, and microtubules. Sorry,
Yeah.
maybe I'm wrong. Even if it is.
But again, if you've knocked out actin and microtubules,
your cell is just dead.
If you
but this is what
you put the cell in hydrogen peroxide, presumably the mitochondria no longer successfully relocate because they're fucking gone.
Well, there's a if you put clammy cells in hydrogen peroxide, you get really big paranoids.
Okay, bad example. Why do they survive? Okay, never mind.
Okay, no. So the thing that I found interesting, or like most interesting about this Miro mutant, is that there's no phenotype. That there's no effect on CCM. And I think, again, this got shoved to the supplementary information, but it's figure S7, which, again, you said you thought should be in the main figures.
Yeah.
But S7D shows a growth assay where 2 mutant alleles of Miro are growing just fine at very low CO2.
So I guess there's
So
maybe they're not successfully getting rid of Miro, or the other thing is this would be a big deal if you were doing it in like a mouse or something, right? But it could be that Miro just doesn't do what we think it does in clammy.
Okay, but the phenotype that they do observe in Miro is that there's no alignment of these microtubules. They do mitochondria. My God, I'm going to make that mistake a million times.
Mitochondria is hard, man.
I'm sorry. Sorry to everybody out there for every time I mess it up. But the mitochondria move to the outside, but they don't form this beautiful aligned pattern. So is that saying that the alignment is kind of like an artifact that isn't actually important for the cell function, and that it comes from the microtubule, like the way the microtubules move the mitochondria?
Yeah, it's an interesting interpretation. I feel like you've thought about this much more critically than I have. I was very quick to be like, hey, if you hit me in the head with a bowling ball and then start trying to study my behavior, I don't think your results are going to be very interesting, regardless of what you find out. That's kind of how I interpret this. Like, if you start being like, oh, he slurred this word, but not this other word. But when I hit them with this other colored bowling ball, he couldn't walk right.
But here, the function is totally fine. You've been hit with a bowling ball and you have these huge gaps in your mitochondrial shell, and now you're growing just fine. So then what's the point of the mitochondrial shell? And this was the same question I asked many years ago, which is is it just an artifact of the cell having to rearrange so dramatically?
The organization, I really don't think so.
Yeah.
That organization looks so much like a circuit board to me. I know you don't see the circuit board thing, but I would say there is a very
what's the benefit of a circuit board?
Periodic length scale there.
Yeah, I think they
and the periodic length scale, I think, must have some biological function.
Okay.
Give me diffusion coefficients for ATP, CO2, and oxygen inside a clammy, and I'll give you a theory on why that length scale would pop out.
We can do that this evening. We can read Ned's paper again. Ned Wingreen and Chen Yi and Alexandra have a beautiful modeling paper where they make all these assumptions. And
I'm going to transition to the next topic.
Sure.
And I'm going to do that with the word mixotrophically.
Okay.
What does the word mixotrophically mean?
So clammy can do multiple kinds of metabolism. They can grow photoautotrophically, which means they're doing only photosynthesis.
Why don't they call that doing photosynthesis?
Trophic. I'm not a biologist, but they also can just take up carbon from the environment and bypass the whole photosynthesis thing. And
that's mixotrophically.
Mixotrophically means they can do both. They can both photosynthesize and take up. So there's acetate around or some other carbon source.
So they do these experiments that essentially show that the mitochondrial relocation correlates with the carbon concentrating mechanism induction. And not necessarily, to me, my reading of this was that the mitochondrial positioning was not very dependent on the outside CO2 conditions. It was really dependent on if the inside of the cell was consuming CO2 or, yeah.
Yeah, so it does. I think that's the correct reading. That what matters is the internal CO2 conditions. So how limiting is CO2 in the cell.
And so I kind of get this thing of there's some optimal place for the mitochondria to be, right? The mitochondria produce CO2, right? And this CCM, they're doing oxidative respiration, they're taking in oxygen, they're producing CO2. It might be best for the mitochondria to be next to the CCM. That seems to be not the case. I was thinking that if you're at the carbon concentrating mechanism, right, and you're trying to produce CO2 to make this respiration thing go forwards, but there's a shitload of CO2 right next to you, you run the risk of running the reaction backwards.
Yeah.
Of taking
so this is what the carbonic anhydrases, I think, are there to try and help with, which is to take things from CO2 to bicarb, which should help both. Yeah.
And this is Michaelis-Menten argument again, right? If you want the mitochondria to be producing CO2 as best they can, the best thing to do is to move it as far away from where the CO2 is concentrated. And that even in a fast diffusion situation, Michaelis-Menten kinetics should tell you that this is still the optimal situation. Because, right, people have shown that parts of the electron transport chain can run backwards, right? And the logic leap from there is that you can risk running the whole thing backwards if you really have too much CO2 and really no oxygen around. Yeah. So one thing I was kind of frustrated with this whole time is we've learned from other papers that there are a lot of really cool ways in CLAMI that you can do OCR, oxygen consumption rate measurements, and CO2 consumption rate measurements through just clever optics, right? That there's 2 photon equivalents of how quickly are you going through photosynthesis and stuff like this. No measurements like that at all, I don't think. What I got was a bunch of beautiful pictures, a bunch of drugs that I don't care about, and a bunch of RNA experiments.
You weren't happy with the O2 evolution experiments?
Okay, so were there O2 evolution experiments? Is this what figure 6 is supposed to be?
Figure 5? Wait, let's see. Figure 6. Yeah, figure 6 is O2 evolution experiments.
Yeah, so this was my one thing. I don't know what these graphs are. What is K-half?
K-half is the affinity for inorganic carbon.
So how do I read one of these graphs?
So at very low CO2, you have a low K-half, which means a high affinity.
And how did they measure that?
They measured that. Yeah, this I was kind of hoping you would know.
I looked at this graph. I tried to make sense of it for a few seconds, and I was like, I'm out. Whatever this measurement is, you got to provide. This is. I'm not going to put more work into this graph than they put into this graph.
Okay. Yeah, I don't this is. I was hoping you would know more about how they measure these things, but they don't say in the main text. They just say that they measured it. The concentration required to reach half maximum O2 evolution capacity.
If this is,
I know this is a standard thing to measure. Some people in Martin's lab have done it a long time ago, but it's not something we do a lot. But I think it's a pretty powerful tool because this is really the proof of this is what carbon concentrating mechanisms do is they increase your affinity for inorganic carbon. So it's I think the most definitive readout for whether your CCM is working.
If this is the most important thing, then it's a pity that they buried it so far.
I'm not saying it's the most important thing. I'm saying it's the most definitive readout for function.
Oxygen evolution measurements. HC, LC, and VLC grown cells were pelleted for 3 minutes at 1,800 G and resuspended in 20 micrograms CHI per milliliter in 2 milliliters of fresh medium. The medium is TP at pH of 7.8 that was sparged with CO2-free air. The cells were then loaded into the sample chamber of an oxygraph oxygen electrode system. The chamber was sealed and the cells were exposed to 300 micromolar photons per meter squared per second. Once the oxygen evolution rate declined to net zero, sodium bicarbonate was added to the suspension at a final concentration of 10, 50, 250, and 1,250 micromolars, and the O2 evolution rate measured. So
chemistry.
The O2 evolution goes to nothing. They add sodium bicarbonate at different concentrations, and then they read the O2 evolution rate again. And they're doing it via an electrode.
Yeah.
The K1/2 was calculated from the fitted curve for each strain. AOA was added at 1 millimolar to the cell culture 30 minutes prior to measurements of O2 evolution. MX and sham were added at 2.5 micromolar.
Yeah, these are the inhibition
these are the
experiments.
inhibition things.
Yeah, the question I had for you also was, do you know what these mix and sham do?
No, never heard of
Okay.
them. I'm sorry. I'm not super familiar with what they're literally doing, but this sounds reasonable. So I was supposed to read these. So a higher K, I still don't know how to read these, man.
Yeah, so lower K means higher affinity for inorganic carbon.
But wait. Why are we learning anything about inorganic carbon here? This was an oxygen measurement.
So you have to use up an oxygen to fix a carbon.
You have to use up an oxygen to fix a carbon. But you can also... What?
Right?
Well, for mitochondria, you have to use up an oxygen to produce a carbon dioxide. And then you're saying the chloroplast... I thought the chloroplast takes in carbon dioxide and produces oxygen.
Okay.
Isn't that what a plant does?
Yes, I'm sorry. you have to produce an oxygen every time you fix a inorganic carbon.
Yeah, it is interesting. This is like a kind of difficult measurement in this system because in a system where you don't have chloroplasts, the meaning of oxygen and carbon dioxide is far clearer. Here, there's some give and take.
Well, so mitochondria, you're saying that mitochondria are also using up oxygen. And so the chloroplast is creating oxygen. And how do you decouple the chloroplast creating oxygen from the mitochondria using oxygen?
Yeah, and I thought that there were these clever mechanisms by which they just used essentially two photon effects where they could be like, look, if the photon that went in gets slightly shifted due to the fact that some of the energy got put into photosynthesis, then we could say that that photon was used for photosynthesis and therefore count specifically what the chloroplast is doing.
Yeah, this sounds like not a standard thing in the clammy field. Read a paper about this?
I mean, did we? Is that one of the ones that we never put out, or is that just like something that I read completely on my own? I remember talking to you about it.
Yeah, I do remember talking about it, but I don't remember if it was that first paper for episode 1 or if it must have been that first paper for episode 1 because there were so many different techniques in that paper.
So yeah, they do a decent job presenting the data. I feel like they could have thought about the data a little bit hard, although if one of the people was retiring in the middle of this, then, never mind. They thought about it as hard as they possibly could have, right? I feel like, again, I'm in a bad mood, but right, these papers are always under the conditions of somebody is about to leave, and here's the compromise paper that we put out so that we at least put out something.
Yeah, I think that's really... Well, so, and they, at the end, figure 7, they do talk about their potential mechanisms or like for why, or there's speculations for why this
super unnecessary. I thought this got super
mitochondria
weird.
could be relocalizing. Well, I think they're trying to be like, somebody follow up on this.
Yeah,
like,
sure.
These are the things that we've thought about. Like, you should think about it too.
But here's how weird it gets. Like, at the end of the discussion, it really feels like I'm reading a proposal that they're trying to get funding for. One promising strategy would involve screening for mutants unable to perform VLC-induced mitochondrial relocation based on single-cell imaging. To this end, intelligent image-assisted cell sorting is being used to develop high-throughput screening of insertional mutants.
Yeah, we don't have to...
They just kind of talk for a little while about, like, what
I wanted to ask you about this glycerate glycolate mitochondrial stuff.
Oh, can I, before I say it, I had mentioned this earlier, Asta, third citation on this paper.
Oh, yeah. This is a very heavily, Ashwani is also cited very heavily. A lot of Martin postdocs have been contributed. Well, okay, before we move on, before I ask these questions, can I say one thing about the discussion?
Yeah.
Which is this, I really liked that mitochondria have been observed to move in other algal species, and that there's evidence of physical interactions between mitochondria and chloroplasts. And so this, I think, is again with the themes that we've been going through in the Lemma Journal Club so far, is plastid mitochondria interactions.
Yeah, welcome to the Lemma Journal Club, where we talk about mitochondria plastid interactions all the time. What do you think is intellectually beautiful about this paper? Or, put another way, what did they discover in this paper?
So they discover that what matters for the mitochondria relocalization is the internal CO2 availability. So if this TAP highlight result, I think, was not known previously. If
you could have had one experiment added to the paper, or, one thing added to the paper.
I really wanted a live movie of the mitochondrial relocalization.
Do you think that is going to be our answer every Journal Club episode? That what we want is live data, live data, live data, live data?
Well, in this case, why not? I'm confused. They put all this effort into making this clover mitochondria, which couldn't they have used MitoTracker for the whole project? And what's the difference between...
I don't think probably anybody knows, except for us, that MitoTracker green, you can just dump it on Clammy and they uptake it. Like, who's tried that before?
Well, I don't know. Ashwani used it after we tried it.
After we told him, yeah.
But I would have tried it. But yeah, wouldn't that be a beautiful movie? They have one live movie of... Did you watch the one...
probably has that movie?
Us. Yeah. Did you watch the one movie they did have?
Yeah, I wasn't impressed.
Yeah.
Did
you watch The Merge? The Merge was more impressive of the bright field and the fluorescence. You can really see that they do pulse with the vacuoles.
What do we think are the deepest takeaways from this paper? What did we technically learn? And how does that move us forward?
Deepest takeaways.
Yeah.
I mean, I still think it's beautiful that these mitochondria form this, like, aligned structure at very when the CCM is induced, and I think it's still an open question is what I learned from this paper as to why they do that.
I think what I've learned from this paper and just kind of the last couple papers that I've been reading in general is that maybe biochemists and biologists have never talked ever. Maybe they've just never encountered each other. Because, like, what is the point of all this Michaelis-Menten in vitro reconstituted length scale reaction diffusion stuff if no biologist has ever considered using it?
Yeah, I think that's a little bit of a inflammatory, provocative statement.
You think I should delete that?
I don't know.
So it's almost time to go get Asya.
Yeah.
Thank you for joining us for another Lemma Journal Club. Please spread this far and wide. We are not very, like, social media people. We tend to boycott a lot of the bigger social media things for ethical reasons, which means we have no online presence. So use your online presence. Send this to everybody you know if you like it, and they'll think that you're really smart because you're listening to this science podcast about mitochondria and plastids and, in the future, other things as well. If you don't like it, give us some feedback on what we could do better. Rate us 5 stars if whatever you're listening to this on has a rating system. And yeah.
Do we have anything else we need to communicate? Episodes on Mondays.
We'll put on some supplementary episodes occasionally. We'll probably put one of those up today. We're going to put up a couple failed experiments on the Patreon for members. This doesn't cost anybody any money, but you do have to become a member to view the failed experiment.
Yeah, only for the die-hard fans.
Yeah, it's not, they're failed experiments, so don't judge us on them. And so we'll put that there, because there are bits of it that we like, and you can kind of see what we're working towards.
Yeah. Yeah, and if you have paper suggestions, let us know.
This recording setup, we will never do this recording setup again.
And thanks for listening.
Thanks for listening. Bye.
Bye.